The overall goal of this procedure is to measure microtubule dynamics in yeast cells.
This method can help answer key questions in the microtubule field, such as how mutations in tubulins and microtubule regulators regulate microtubule dynamics in a living cell. The main advantage of this technique is that living yeast cells are stabilized and imaged for either high time resolution for 10 minutes or low time resolution for several hours. Demonstrating the procedure will be Colby and Cassi Estrem, 2 graduate students from my laboratory.
The yeast cells used in this experiment have been constructed to constitutively express GFP-labeled tubulin. Inoculate about 1 microliter of yeast cells from a single colony on a plate in 3 milliliters of synthetic complete medium. Allow the cells to grow overnight at 30 degrees Celsius with shaking at 250 RPM. On the following day, dilute the saturated culture to an OD 600 of less than 0.1 in the medium. Return the diluted culture to the 30 degrees Celsius shaker for 3 to 4 hours or until the OD 600 is between 0.4 and 0.8. Begin the preparation of flow chamber slides by cutting a sheet of paraffin film into a piece measuring 4 inches wide and 7 inches long. Place a standard microscope slide lengthwise on the 4-inch width of paraffin film and wrap the film around the slide 3 to 4 times such that the slide is covered in film that is 3 to 4 layers thick. Press the layers together to ensure that there is a reasonable seal, but avoid pressing too hard or causing the film to wrinkle. Using a clean box cutter razor and another slide to provide a